In this short video, I present a paper titled Monitoring the Advancements in the Technology of Artificial Cells by Determining Their Complexity Degree. Hints for Complex Systems Descriptors. Written by me, working at the Department of Chemistry, Biology, and Biotechnology of the University of Perugia in Italy, and Professor Pasquale Stano, working at the Department of Biological and Environmental Sciences and Technologies of the University of Salento in Lecce, Italy. In recent years, an important momentum has characterized artificial cell research from the viewpoints of technical, conceptual, and functional achievements. Although the ultimate goal of artificial cell research, which is the construction of minimal living artificial cells from scratch, is still not so near, the aforementioned advances raise an intriguing question. Is it possible to monitor these advancements by measuring artificial cell complexity? The formal definition of complex systems and a rigorous methodology for determining their degree of complexity are challenging tasks not yet accomplished. Despite the lack of consensus on definitions and metrics, it can be agreed that any complex systems can be generally described as a network whose constitutive components are nodes and links. The nodes are the elements of the network, whereas the links are the relationships among them. The network is intended as a highly dynamic because complex systems are maintained constantly out of equilibrium in the thermodynamic sense. The strong interconnections among the nodes confer the power to show emergent properties to the network. Emergent properties belong to the network as a whole. They are collective and pop up through the nonlinear integration of the nodes' features. Any complex systems can be analyzed hierarchically through the reductionist, mesoscopic, and systemic approaches. The reductionist approach requires the determination of all the nodes and links that are the fundamental ingredients of any complex system. In a cell, the nodes are the molecules, and the links are the chemical reactions among them. The complexity of biological cells and artificial cells will depend on how many molecules and reactions are present. An analysis of a cell at the mesoscopic level entails revealing the network modules. There are, for example, signaling, genetic, and metabolic modules within a living cell. Then the complexity of an artificial cell can be determined by evidencing the number, types, and the connection of modules, as well as their functions. Finally, artificial cells can be investigated at a systemic level. The systemic approach analyzes the functional features of the network as a whole. If we embrace the rationale of natural computing, living cells and artificial cells can be conceived as computing systems. Therefore, it's valuable to determine the inputs it receives, the outputs it generates, and the corresponding computations. The larger the number of computations, the higher its complexity. As a case study, in this paper, we analyzed the artificial cell implemented by Noirot and Libchabet in 2004 and published in the article you can read here. We analyzed this artificial cell at the reductionist, mesoscopic, and systemic levels. Why is it worth measuring the complexity of artificial cells? Measuring artificial cells' complexity can help guide the experimental efforts toward higher complexity, corresponding to more functional systems. Alternatively, complexity metrics can help to minimize complexity given a specific target behavior. Finally, it will contribute to finding a still missing universally accepted definition of complex systems. So thank you for watching this video. And if you want to know more details, I invite you to read our paper by clicking the link in the description below.